A very good morning and afternoon to everyone here virtually. Welcome to today's webinar, LiquidArray, Next Generation Multiplexing for Routine Clinical Microbiology. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Bruker Microbiology and Diagnostics. To learn more about our sponsor, please visit Bruker.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and simply click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well. I would now like to welcome our speaker, Dr. Jennifer Dougan, Head of Business Unit Syndromic LiquidArrays, Bruker. Dr. Dougan, you may now begin your presentation.
Thank you for the introduction and thanks for joining the webinar today. My name's Jennifer Dougan and I'm Head of the Business Unit here at Bruker Microbiology and Diagnostics for our Syndromic LiquidArrays. And I'm delighted today to be introducing LiquidArray to you, our next generation multiplexing technology and its applications in routine clinical microbiology. So today I will start by giving a very brief introduction to the molecular diagnostics business area within Bruker Microbiology and Diagnostics. And I'll then introduce LiquidArray and talk you through how it works and what the diagnostic approaches are with the technology. And then move on to some examples of the clinical applications that we already have available for Mycobacteria, syndromic panels for sexually transmitted infections, and gastrointestinal infections. So Bruker Microbiology and Diagnostics is now a rather broad division, obviously originally with bringing the MALDI biotyper to clinical microbiology for routine microbial identification. Bruker have since expanded in this area to offer solutions to the lab for antimicrobial susceptibility testing and microbial strain typing with our infrared biotyper, and along with our molecular diagnostic solutions, and most recently with our LiquidArray syndromic panels that I will tell you more about today. And for all of our developments, we really aim to keep the patient in focus by bringing technologies to the lab so that you can turn around your lab results in a faster and more cost-effective way. So molecular diagnostics here at Bruker and the products that we offer actually started quite some time ago, back even in the 90s, with Hein Life Sciences developing the genotype assays that work with PCR on a strip, and then evolving throughout the years to develop real-time PCRs, and in 2018 were acquired by Bruker, and then together we brought the innovation of LiquidArray into the market in 2019, and then this year launching LiquidArray with applications for syndromic panels. So our full portfolio of solutions includes extraction systems, extraction chemistry, detection systems, and IVD assays across a broad range of clinical areas that you can see here, ranging from mycobacteria all the way to human genetics. An example here of their FluoroCycler XT and the validated assays that run on that show the breadth of the portfolio that we already have available on that instrument. And this is really a high-performance thermocycler that I will talk more about today that does not only real-time PCR and melting curve analysis, but also powers our LiquidArray assays. So what is LiquidArray? Well, it's a next-generation multiplexing PCR-based technology. It provides comprehensive coverage for infectious disease diagnostics, and it does it in a familiar PCR-based workflow. As said, it's only possible with the unique features of our high-performance FluoroCycler XT, and I'll come on later to talk about them in more detail and how it is that they support the LiquidArray technology. And all of our assays running LiquidArray are automatically interpreted and are provided with results at a glance, bringing then next generation multiplexing for infectious disease diagnostics really to the routine clinical microbiology laboratory. I'll now talk about what actually happens when we're running a LiquidArray assay and how it delivers next generation multiplexing. So as said, it's a PCR-based technology, and we're using linear after exponential PCR with lights on and lights off probes. It's underpinned by the features of the FluoroCycler XT, and by that I mean the well-to-well thermal precision of the instrument, as well as the outstanding optical alignment. And using the integrated software, FluoroSoftware XT, we're able to provide results at a glance. So looking at this in a little bit more detail, the linear after exponential PCRs and asymmetric excess PCR with limiting and excess primers. So looking at a target strand of DNA here, doing normal PCR cycles. After multiple cycles, of course, we all know that you generate many copies of PCR product. But we also here have a limited primer, meaning that after the exponential phase, we're generating single-stranded PCR product. And that really drives high sensitivity by the generation of this single-stranded DNA. Looking more closely then at the PCR product and how it is that we go about detecting that, we're using lights on and lights off probes. So using our lights on probe, we have a fluorophore and a quencher, and this I'm sure will be familiar to many of you, that when the fluorophore and quencher are kept far apart, such as when the probe is bound to a target piece of DNA, the fluorophore is able to fluoresce and be detected by an instrument. Whereas when the probe is not on the target strand of DNA, then the fluorophore and quencher come closer together and you no longer would see that fluorescence. Our lights off probe has a quencher only, and I'll show how this works in practice. At low temperature, we would expect no fluorescence with the lights on and lights off probes designed to bind to the target adjacent to one another. So the lights on probe is directly next to a lights off probe. The fluorescence is quenched, and so therefore no fluorescence is detected. As we increase the temperature, we expect fluorescence to be detected. The lights off probe is designed to be released from the target at a specific temperature. So when this happens, of course, we can then see fluorescence from the lights on probe. As we raise the temperature further, we would see fluorescence switching off as the lights on probe has been released from the target and you no longer see fluorescence. Of course, we design our probes such that these binding and release events happen at predictable temperatures, and you then are able to see fluorescence against temperature and interrogate the different probes at different temperatures. So we have a temperature-dependent multiplex detection within each optical channel. As said, this is only possible on the FluoroCycler XT because of that well-to-well thermal precision, because we're really looking at multiple binding events in the one fluorescence channel across different temperature steps. We need to have very highly accurate and precise temperatures reached within the wells, and we can achieve that with the FluoroCycler XT. We also in the instrument have outstanding optical alignment, so not only can we multiplex within one optical channel by using temperature, we're also able to multiplex in a more traditional way across the optical channels. And in our case, the optical alignment means that there is no crosstalk and we require no color compensation. So we're giving multiplex detection across those 5 optical channels with no crosstalk. And as you might have seen, you of course then can generate these rather unique fluorescence signatures. And so it's really important that we bring this to the routine lab with the integrated Fluoro software to provide then results at a glance. You will see the fluorescence signatures produced in the report, but it's not required for you to interpret them. That's done already by the integrated software, giving an unambiguous report. Automated results interpreted, and machine learning algorithm providing results at a glance. So we apply this technology in a couple of different ways. And in the first example here, for multiplex pathogen detection, we can probe multiple different pathogen sequences using lights on probes, each of them, of course, with a different melting temperature, so that we, in the same optical channel, can detect multiple different pathogen sequences. And we can also apply it to multiplex detection within a region or regions of a single target. So that's where, again, with different melting temperatures, we're able to multiply probe a single sequence with the lights on and lights off probes. And with multiplex detection using multiple probes within a single target. And I'll show that in a little bit more detail. So here we have a target strand of single-stranded DNA, and showing here the lights on and lights off probes that have bound to that. So as we apply an increase in temperature, we can watch what happens with the probes, and also I encourage you to keep an eye over here on this fluorescence against temperature plot. So as the lights off probes are desorbed, you get an increase in fluorescence as the lights on probe switches on. And we see this happen across the region that we are looking to detect. Then as we increase the temperature further, the lights on probes now come off of the target, and we see then reductions in the fluorescence that we are detecting. And this is all done in a predictable order on the basis of how we have designed those probes. So we know the temperature that they will desorb from the single-stranded target. Now, if we were to have a mutation in the target sequence within this probe region, the melting temperature of that probe would change, and as such, the fluorescence signature would also change. And you can see quite clearly then that you could identify the difference of a wild type from a mutation. So with LiquidArray, we're really able to target multiple targets per optical channel. We generate these unique fluorescence signatures. We have high resolution between the fluorescence signatures, and we can use up to 5 optical channels with no crosstalk between them. So this multiplexing within each channel, plus the multiplexing across the 5 optical channels, is what really delivers the next generation multiplexing that we see with LiquidArray. We offer multiple workflows depending on the clinical application, and that would range from manual extraction, our smaller extraction system that extracts 12 samples in a run, to our semi-automated GenoExtract XT system, which can process up to 96 samples and set up your PCR plate. Then the assays are run on the FluoroCycler XT, and as said, the integrated software provides results at a glance. So looking then at the applications here for within mycobacteria and resistance detection and differentiation. So as an introduction, tuberculosis is a disease caused by Mycobacterium tuberculosis. It's an infectious disease with a tremendous public health impact, with approximately 10 million new cases per year. and around 500,000 cases of drug-resistant TB worldwide. And there's also non-tuberculous mycobacteria. So these are environmental bacteria that can cause disease, and the incidence of non-tuberculous mycobacteria are increasing worldwide, with treatment of NTM disease dependent on the species, and also detection of NTM to the species level can provide then important clinical and epidemiological information. So there are two questions, I guess, clinical questions in the mycobacteria space that we're looking to answer here. So FluoroType MTBDR 2.0 is powered by LiquidArray, and it provides detection of the Mycobacterium tuberculosis complex and its resistances to rifampicin and isoniazid from patient specimens or culture, and detects up to 45 different mutations in the Mycobacterium tuberculosis complex, and is WHO endorsed. So here you can see quite clearly from the report that we would see from LiquidArray that yes, there are fluorescent signatures there, and the reporting will tell you whether the MTB complex has been detected. It will tell you the mutations that have been detected, and whether actually the organism is whether the sample is showing mycobacteria that is sensitive or resistant to the drug in question. And we're covering up to 45 mutations in the MTB complex in a single well, which is something normally only achieved with sequencing. For FluoroType mycobacteria, we are detecting and differentiating 32 clinically relevant non-tuberculous mycobacteria and detecting the Mycobacterium tuberculosis complex in one well from positive culture. So again, here really the power of LiquidArray is shown. You can detect 32 of these NTMs and the MTB complex and Mycobacterium genus in a single well. And this is just here showing the range of NTMs that we're covering with this assay. And again, the FluoroCycler XT report that you would receive after having done your run unambiguously tells you which NTM has been detected by the assay. So with these LiquidArray assays, Bruker's expanding the comprehensive portfolio that we already have for mycobacteria testing to include this first-line resistance testing of 45 mutations in a single well, which is being achieved by the LiquidArray technology. And we have a second-line resistance assay with LiquidArray MTB XDR coming in 2023. And we have our assay providing differentiation of these 32 different NTMs in a single well with FluoroType mycobacteria. In all cases, the fluorescence signatures are fully interpreted by the integrated software, providing you results at a glance. And so we're really delivering a workflow that's compatible with routine testing, with reflex testing from the same isolate possible. So from the same DNA isolate, you can test for TB and your multi-drug resistance, as well as the second-line resistance, or the second-line resistance assay coming soon. And also the genoextract isolate is suitable for sequencing if that's something that you also need to do for other research purposes or doing some epidemiological studies. In terms of LiquidArray for our syndromic panels, I'm introducing here our syndromic panel for sexually transmitted infections. So STIs are caused by a broad range of different bacterial, viral, and parasitic pathogens. The top two most common bacterial pathogens in Europe being Chlamydia trachomatis and Neisseria gonorrhea, of course. Trichomonas vaginalis and Mycoplasma genitalium are also recognized as important and under-recognized causes of STI. And in 2020, WHO estimated 374 million new infections with one of the four STIs, chlamydia, gonorrhea, syphilis, and trichomonas. And it's really important that we are detecting STIs. Untreated, these pathogens can lead to many complications from chronic pelvic inflammation, through pregnancy complications and infertility, vaginitis, cervicitis, and urethritis, and often they may be being carried without symptoms. And so it's really important that we are identifying people who are carrying these, and also to be able to provide the correct treatment in cases as well. So FluoroType STI detects 7 prevalent bacterial and parasitic STI pathogens in one well. That includes dual target detection of Chlamydia trachomatis and Neisseria gonorrhea, and were validated from urine and genital swabs. So we've developed this as a syndromic panel for routine testing, covering the 4 most prevalent pathogens, plus 3 potentially pathogenic species as well. It's a qualitative test, and as said, the sample materials validated are for urine, genital swabs, endocervical, vaginal, cervical, and urethral. So we have a dual gene target strategy for both Chlamydia trachomatis and Neisseria gonorrhea. The 2 targets for Chlamydia trachomatis ensure that we have full coverage across cryptic plasmid and genomic strains, but only 1 target is needed to get a positive result. So if you detect one or the other target sequence, you would get a positive result. Whereas the 2 gene targets for Neisseria gonorrhea are serving to increase our confidence that we can report with a Neisseria gonorrhea positive. Because of the frequency of gene recombination between Neisseria species, it's really important that we have a stringent test, that if you have a positive result, you can be confident that it is Neisseria gonorrhea. And so for that reason, we require both of these regions in the POR8 and the OPA genes to be positive in order to report a Neisseria gonorrhea positive, so that you can have confidence in that result. A single positive would report for Neisseria species. Here's an example report of what you would see using the FluoroType STI assay. And again, you can see here an example where 2 genes of Neisseria gonorrhea are detected and we're reporting Neisseria gonorrhea positivity. And in this example, a single Neisseria gonorrhea gene is detected. And here we're reporting Neisseria species. The additional organisms, Mycoplasma hominis, Ureaplasma parvum, and Ureaplasma urealyticum, are commensal organisms, and so we do report these out, but the results for these species should be clinically interpreted in the context of patient presentation and in the absence of other causative pathogens. The FluoroType STI is currently validated on our GenoExtract system onto the FluoroCycler XT and providing results at a glance with the Fluoro software. And it will be available soon also on the GenoExtract FlexT, our larger liquid handling system. Showing some results from our performance evaluation studies, you can see here really very excellent sensitivities and specificities and very low rates of invalid results that we're seeing. These results shown for genital swabs, and then here for urine samples. Again, very low rates of invalid results from testing urine samples, and also a low number of indeterminate milk curves. The performance evaluator for FluoroType STI it gave the following quote, that based on the performance in their laboratory, the use of FluoroType STI to detect sexually transmitted pathogens in the daily routine of a service of microbiology is highly recommended. And I think really that comes down to the confidence in Neisseria gonorrhea reporting achievable through the dual gene target detection, the great analytical and clinical performance, and that the workflow is compatible with daily routine, and the ease of achieving those results at a glance with the integrated software. Looking now at our next syndromic panel that was just launched this summer for gastrointestinal infections. So gastroenteritis, of course, is an inflammation of the lining of the stomach and intestines, and we know characterized by diarrhea or vomiting. And there's many causes. It can be viral, bacterial, parasitic. Can cause life-threatening diseases, especially in children, immunosuppressed people, and the elderly. And of course, here the incidence range of diarrhea in children in Europe. Most cases do resolve within days, but persistent of severe cases can of course require hospitalization. And it's a major cause of hospital admission and has a large economic impact. And traditional diagnostic approaches, including microscopy, culture, and PCR, are labor and resource intensive, and also take a certain amount of time to get results back to the clinic as well. And there are different workflows basically for bacterial species, parasites, and viruses, and even for within different bacterial species. The UK standards for microbiology investigations from Public Health England indicate that the use of multiplex panels, testing by syndrome, can simplify routine service through rapidity, the reduced costs, reduction of unnecessary antibiotic usage when compared with single testing. And so we developed LiquidArray gastrointestinal to detect up to 26 pathogens that cause gastroenteritis in 2 wells. It's a single test for bacteria, bacterial toxins, viruses, and parasites, and we provide it in a streamlined workflow from stool sample to result. So obviously the bacteria, viruses, parasites, and pathogen-associated toxins are all detected in a single protocol, and we're covering 26 of them across these 2 wells, with well 1 detecting the most prevalent pathogens. So here listed your bacteria and toxins, so the viruses of norovirus and rotavirus, and your parasites. In well 2, we are reporting and detecting the less frequently reported pathogens, again, bacteria, viruses, and parasites. This is validated on stool samples. You can see again an example report here with showing the Fluoro software, again providing these results at a glance. So yes, you will see traces, fluorescence traces in your report, so you will be able to see the primary data. However, you will be given a completely unambiguous report of what organisms have been detected, or for example, if all were negative, and also an interpretation of your control's validity. Workflow for LiquidArray gastrointestinal starts, of course, with the stool sample, which is swabbed and taken into stool buffer, and then vortexed, briefly centrifuged, and then the actual stool buffer sample is taken into the GenoExtract XT along with the other reagents. And in the GenoExtract XT, the instrument will automatically extract the samples and then set up your 2-assay well PCR. The PCR plate is centrifuged, it goes into the FluoroCycler XT, and at the end of that LiquidArray cycle, you get results at a glance with the software as previously shown. Showing here then some results from the performance evaluation. With the PCR1 targets, we see very good sensitivities and excellent specificities across these targets, and very low rates of invalid results, especially when you consider testing from stool samples. 0.2% for invalid results was really very impressive. Our evaluator was very happy with that. And looking at the less frequent pathogens in well 2, you can see here again really very good sensitivities and specificities, and low rates again of these invalid results. This is really important, of course, for when you are running high numbers of stool testing and looking to really optimize your laboratory workflow, to know that you would have confidence in both specificity and also lack of invalid results that can hamper other tests. So with LiquidArray gastrointestinal, we're providing comprehensive coverage with 26 pathogens simultaneously detected across this single protocol for bacteria, viruses, parasites, and pathogen-associated toxins. The 2-well format, covering all of these pathogens in only 2 wells, means that we are really compatible with that daily routine testing. I speak to many customers who have to test over multiple wells and multiple tests with their current system, and looking to bring that in where they're only having to run a single protocol with 2 wells means that they can meet the needs of the clinical partners that they're serving. And of course, results at a glance provided. As a summary then, across our LiquidArray assays, we're providing comprehensive coverage where labs can replace multiple tests with one, providing workflow efficiencies and fast results to the clinic. It's performed in a familiar PCR-based workflow, which is then easy to implement in the routine laboratory. And we're providing infectious disease testing for syndromic panels for gastrointestinal, as you've seen, in sexually transmitted infections, as well as mycobacteria and resistance detection and differentiation. Shown also here that we've developed this with compatible nucleic acid extraction kits and workflows, taking you from patient sample to results with very high quality. And obviously providing these results at a glance in a user-friendly report so that you can report accurate results with confidence and without any need for user interpretation. So today I've shown you the power of next generation multiplexing across our portfolio, with Mycobacteria assays achieving 45 different mutations detected within 1 well with fluorotype MTBDR 2.0, and up to 32 different NTMs detected in 1 well with fluorotype Mycobacteria. Our fluorotype STI syndromic panel is detecting 7 pathogens in 1 well, and our LiquidArray gastrointestinal is detecting up to 26 pathogens for gastroenteritis across 2 wells in a single workflow. We also have a LiquidArray in development in respiratory. We're really then covering infectious disease diagnostics with comprehensive coverage per assay, providing streamlined PCR workflows for you with results at a glance in order to really help increase efficiencies in the routine clinical microbiology laboratory. So with that, I would like to thank you for your attention, and I'll open now for any questions.
Thank you, Dr. Dougan, for your informative presentation. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have any questions you would like to ask, please do so now. Just use that Ask a Question box located in the far left of your screen, and we will answer as many of your questions as we have time for. Okay, we already have some great questions coming in. Dr. Dougan, let's start with this one. With the enhanced multiplexing capability within the fluorescence channel, do you find that this has a negative effect on specificity?
Thanks, Christina. Yeah, that's a good question. It's something that I think many people are concerned about when they look at a multiplex test, but it's not what we've seen through our performance evaluations. So when we look at the data, the specificities that we're seeing with our STI assay, for instance, are in the range 95 to 100%. And looking at gastrointestinal, the specificities they are clinically tested are 98 to 100%. So we don't see a negative impact on specificity.
Thank you. And our next question. How do you deal with new resistances and variants within your LiquidArray assays?
Yeah, great. Another good question. So we do get asked this. I get asked this quite frequently. So basically, we keep a close eye on the literature and on surveillance programs to keep up to date with mutations as they emerge and are reported. As an example, with the fluorotype MTBDR assay, that assay is probed extensively in the region of the genes that typically show mutations. And so a new mutation in the probed region would generate a different fluorescence signature. The software would automatically determine that it was something new and not expected, not one of the typical or known mutations, and it would report as indeterminate. So we welcome interaction and collaboration with our customers if they do see such cases to get in touch and work together with our R&D team where we would look to understand and where relevant incorporate any such strains and any such results.
Dr. Dougan, do you plan to include or produce a kit for M. genitalium resistances?
So Mycoplasma genitalium resistance is a topic that we are looking at, along with some other possibilities around what will be next for our SDI portfolio. So it's not something that we have currently in the assay or immediately in development, but it is an area that we are looking to as we look to expand and grow our solutions for the lab.
Thank you. Now, have you looked at additional sample types with the SDI assay? For example, rectal or throat swabs?
Yeah, I mean, depending on the population that's being tested, these can be really very relevant sample types. Currently, our assay, as shown today, is IVD for urine and for genital swabs. We have tested with rectal swabs and with throat swabs, and we know that the assay does work with those sample types, and we would look to incorporate that within the IVD at some point in the future.
Dr. Dougan, for the LiquidArray gastro test, with so many targets reported, do you see positivity in most samples? And how does this relate to single pathogens versus co-infections detected?
Yeah, another good question, similar, I guess, to the questions that we receive around specificity. So I think, again, there's some concern that, or some assumption that with so many parameters covered by the LiquidArray gastrointestinal, that you would see basically positivity in all samples or something like that. It's not what we see in practice. Through the performance evaluation studies where the samples were tested as part of sort of routine workflow, we saw positivity in 35% of those samples. So 65% of samples were all clear, all negative, not positive for any of the pathogens. And of the second part of that was single pathogens compared with co-infections. So of those 35% that are positive, we saw single pathogens in the majority, about 85% of the time, and co-infections about 15% of positive samples, which is significant in that, obviously culture, often, one organism can outcompete another, and so we may be missing co-infections, or we may not even the lab may not be testing for those organisms at this time. So it does provide for a broader and more comprehensive coverage with the lab running this test.
Thank you, Dr. Dougan. And we'll wrap with this final question. Given the inhibitory nature of stool samples, fecal material, what are the typical invalid rates?
Yeah, so as shown earlier in the presentation, Another concern around fecal material, as I think the questioner is getting to, is you can often see quite high numbers of invalid rates with other tests. Our invalid rate was around 0.2%, 0.6% for well 1 and well 2. So we are really seeing very low levels of inhibition and invalid results, especially given that we're working with complex sample media.
And Dr. Dougan, I'm going to squeeze in another question here. How much training do you need to switch to running LiquidArrays?
Oh yeah, that's a great question. So basically, if you can run PCR, you can run a LiquidArray. I mean, we've developed it in such a way that, yes, it's giving you next generation multiplexing, really advanced level results. We're able to deliver that, but we do it in a way that doesn't require, any special training beyond what you would be used to using, nor it's set up in a normal, familiar PCR-based workflow and doesn't require, expensive cartridges or anything like that. So it's very reliable, very robust, and very easy to use. And obviously, again, I know I've mentioned it a few times, but we give all of those, you get all of those results with the unique fluorescent signatures. We're getting a lot of power out of that information, but the user doesn't need to take time to learn that or to interpret it themselves. It's fully interpreted in the system, and you're provided the results unambiguously. It tells you what target was detected, whether any targets were detected, whether your controls worked. The whole thing is fully controlled.
Dr. Dougan, thank you so much for your time today and for your important research. Do you have any final comments for our audience?
Thanks, Christina. No, I would really just like to thank those for joining today. It's been an absolute pleasure to introduce LiquidArray to you, and I look forward to more in the future where we are able to highlight further developments in this field. So thank you.
And thank you again. And we'd also like to thank LabRoots and our sponsor, Bruker Microbiology and Diagnostics, for underwriting today's educational webcast. Before we go, I also want to thank our audience for joining us today and for their interesting questions. I want to remind you that any questions that we didn't answer today or any of those coming in during the on-demand period, they will be addressed by our speaker via the contact information that you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us, and take care. Bye-bye.